Okay, so this is going to be water transport in plants, basically how water flows through plants. And we're going to start with just some basics. I'm sure you've heard this in basic biology, but basically how molecules move through solutions of fluid is you have a solvent is the liquid in which things are dissolved, and those substances may be salts, electrolytes, or sugars, or other things. And then there are semi-permeable membranes, of which the lipid membrane of most cells, which allows some things in and at a quicker rate than other things. And so that's what we have illustrated here. And what happens is, in diffusion, molecules move from a greater concentration to a lower concentration. So these blue molecules, if you remove this barrier, will move from where they're all concentrated into this area where they aren't, and the red ones as well. They will go in opposite directions until they are evenly spaced. Okay. Here we have two hypertonic solutions. One is hypertonic with red molecules, one is hypertonic with blue molecules, and then they're going to even out until they are all isotonic, meaning they have the same amount of solutes in each solvent. Osmosis then is the diffusion of water through a semi-permeable membrane, and again is based on evening out these molecules to where they're all evenly dispersed. Okay, so osmosis then creates a pressure, so the desire of water to flow one way or another can push against a membrane or can push against the cell wall in the case of plants. So osmotic pressure is the pressure required to prevent osmosis. So it may have a physical barrier or something which is going to prevent osmosis. It must be at some strength to prevent that. The osmotic potential balance by resistance of a cell wall. So you have pressure potential, or turgor potential is the potential. Sorry, is the pressure that develops against the wall as a result of water entering the cell and wanting to leave. So here you have it in a hypotonic solution, meaning there are more solutes within the cell than outside of the cell, and so the water wants to come in. Okay, and the water will come in and fill that central vacuole, and it will cause this cell to swell, and it'll be counterbalanced between the properties of the cell wall, which will hold it together. So that's a turgid cell, one that is firm due to that water gained. Excuse me. And then if you do not have enough osmotic pressure, the cell will become flaccid or loose, no longer taut and tight on the edges. Okay, so that will happen if you are in an isotonic or a hypertonic solution, where the water going in will match the water coming out, and so there's nothing to push against the cell wall. The central vacuole will not push against it. So the water potential of a cell then is the osmotic pressure plus the pressure potential, the turgor pressure. Okay, and water moves from a cell with higher water potential to cell with lower water potential. So what will happen is if you have too much water leaving the cell, the protoplasm will be pulled away from the cell. So the outer part of the cell will get pulled away from the cell wall. So here's a normal cell, and these are plasmolyzed cells. So you can see the central vacuole has reduced, and all the cell parts are pulled away from the cell wall. So water must go through a plant, a multicellular organism, through these powers of diffusion and osmosis. So it enters from the soil, water enters from the soil through intercellular spaces of roots and root hairs. So it's coming then through there, just by osmosis. And then it must pass through the Casparian strip, that barrier in the root, which is a selective membrane, so it only lets specific things in with the water. So we'll go through the Casparian strip, and then we'll go into the xylem. From the xylem, it will go into the stem and the leaves, and then into the veins of the leaves, into the part of the cell where the mesophyll is. We'll fill that up, and then it will diffuse out through the stomata through the process of evaporation. So there is another process in seeds where water will basically impregnate or imbibe the cells, imbibe the seeds. So what happens here is cellulose and starch develop an electrical charge when they are wet, and that will cause water then to diffuse into the seed, and those water molecules will then adhere to other larger molecules and result, negatively charged proteins, and that will result in the swelling of the tissue. So you see some dry seeds and then some imbibed seeds here. This is one of the processes of germination. So before the seed can grow, many seeds will have to be imbibed first. So osmosis and diffusion are passive transport, meaning they do not require ATP. Active transport, on the other hand, is where we move substances from low to high concentration, so against where they want to naturally go, or against the gradient. And that requires ATP, or requires energy. And one of the ways it does that is through a process, an enzyme embedded within the lipid bilayer called the proton pump. Proton pump uses ATP to send protons through to a higher concentration. Okay, and transport proteins, again, which are embedded on the lipid bilayer, are important for transferring these solutions or solutes outside and inside of the cell. So what fuels the water movement through the plants is first transpiration, which is basically just evaporation through the stomata into the atmosphere. 90% of the water that a plant absorbs through its roots is eventually transpired through the stomata. Water is needed for cell activities, like we mentioned before, the shape of the cell, and important for evaporation, for cooling. So what plants will do if there's too much water loss through stomata, though, is they will close them so that you can no longer have water being evaporated out of them. Now, the process by which water moves through the plant is called the Cohesion-Tension Theory, or hypothesis, which is basically that transpiration generates tension. So as that water is pulled out of the air spaces of the mesophyll, it will create a negative water pressure, in which the water will then want to flow from the veins of the leaf into the mesophyll. Okay, and then from those veins, or from the water column, from the xylem into those veins. And so what's holding the water in these xylem tubes is adhesion to tracheids and vessels. So we see that here, where they're forming hydrogen bonds between the walls of the tracheids and vessels. And then also the water will stick to itself through hydrogen bonds, and that is called cohesion. Okay, so by those two processes, adhesion and cohesion, you have a water column, or a continuous bead, or it's kind of like a straw filled with water. So what happens is water evaporates from the mesophyll cells, and this creates a lower potential than the other adjacent cells. Then water will flow from that greater to lower water potential. The water from the veins will then flow into the mesophyll. The water from the xylem will then flow into the veins all the way down to the roots, which will flow into the xylem. And the soil will then, water from the soil will flow into the roots. This is also aided by osmosis, so by having a greater concentration of solutes in plant tissues, the water will want to follow that concentration of solutes to even out the concentration. So stomata, the opening and closing of them, will regulate transpiration. If they are closed, then there's no evaporation occurring. And it does that through its stomata, which are formed through 2 guard cells and 1 stoma, 1 opening. And transpiration rates are also influenced by humidity, light, temperature, and carbon dioxide concentration. So transpiration is also regulated by other processes, including photosynthesis. Okay, and stomata have to be open for photosynthesis to occur, at least the capturing of carbon dioxide. So the first step, to open the stomata, guard cells will actively transport potassium ions from adjacent epidermal cells into their cells. So you get all these ions into the cell, potassium, one of them. And that creates a lower potential in guard cells, and then water will then follow those solutes into the guard cells via osmosis. And what happens then, it fills up with water, it becomes turgid, those vacuoles fill with water, and that causes the shape to change into a open position. Stomata close when photosynthesis is not occurring, so they don't lose water that way. And so to have it closed, you have potassium ions diffusing out of the guard cells. Water will then follow it because of the water potential, and then guard cells become flaccid and stomata close. So here's a flaccid stomata here. So there is some, as water flows through xylem out from the roots up to the leaves, there is another process by which water will flow, not fueled by transpiration. And it usually happens if you have a cool night followed by a warm, humid day. And then you get water droplets, which are produced through hydrothodes at the tip of the veins. And when at night, what happens is pressure in the xylem, element forces, so cohesion and tension, and osmosis will cause the water to be pushed out of these small pores called hydrothodes, and that leaves these drops of water on the plant. Okay, so that whole process is called eutation. Now, most plants are C3 plants, and they will leave their stomata open in the daytime and close at night, so photosynthesis occurs in the daytime. However, C4 and CAM plants have alternate processes. What they do is they store carbon dioxide in the form of organic acids. They make these 4 carbon chains, and they do that in the daytime so that at night they can access these organic acids and then finish off the Calvin cycle and make the carbon dioxide into sugars. Okay, and then the difference between C4 and CAM plants: C4 open in daytime and close at night. CAM plants open their stomata at night and close in the daytime. So there's no water lost in the daytime and a lot less at night. Okay, so the movement of water occurs through transpiration. The movement of sugars or organic things occurs through a process called the pressure flow hypothesis. So what happens is organic solutes, and we'll just refer to them as sugars, flow from the source to sinks. Okay, and then it's utilized as food, and then water exits. So what you do is you have a source in the summertime when you have photosynthesis occurring in the plant. You should have lots of sugars being made in the plant, or the leaf, and so that will be the source in this example. Okay, so we have our leaf, and what happens is sugar is first actively transported into sieve tubes. Okay, this is phloem loading. And then because that is now a greater concentration of solutes in there, water will follow it by osmosis and try and even things out. Well, now that you have a greater concentration of this watery substance with lots of sugar, that will want to even out through the phloem, and so water will naturally flow from this source into a sink, an area where they need sugar. Okay, so let's say this is the stem or the root, somewhere where you want growth to occur. All right, sugar is then actively removed from the sink and put into starch or cellulose products, so it's no longer sugar, and that causes more, that reduces the amount of sucrose in the sink and causes it to continually flow into there. Now, when you have excess water, that water will flow into these xylem vessels, so that will get rid of the sugar, get rid of the water, and allow a continuous movement of this pressure flow. So mass flow occurs from higher pressure at source to lower pressure at sink, so that's what we have going on.